Thank you very much. It's a pleasure to be here today. My presentation is going to be about hormesis, its biological foundations, and its therapeutic implications. Let's start with the definition of hormesis. At least this is my definition. The way I see hormesis, I see it as a dose-response phenomenon that's characterized by a low-dose stimulation and a high-dose inhibition. It's a non-monotonic, biphasic dose response with very specific dose-response features. Now, in the world of toxicology and pharmacology, we tend to deal with really two basic dose responses, the threshold model and the linear model. The threshold model has a very long history, preceding the linear by far. And the threshold model has been used by toxicology and pharmacology in their hazard assessment, in their efficacy assessment, and it basically has been the model that has been used to set so many different environmental exposure standards, safety standards by food and drug associations across the world. It's the standard model. It was challenged in the 1950s with respect to its capacity to deal with carcinogens. And in a major policy development, the U.S. Academy of Sciences Biological Effects of Atomic Radiation Committee in 1956 recommended a switch from the threshold model to the linear model when dealing with mutations, things that could affect the genome. Within a year after that, other national committees and international committees decided not only to accept that linear model, but to generalize it to somatic cells, so that it then essentially could relate to the concept of cancer. And today, what we have in essentially every country in the world, you have a dichotomy of models. You have a linear model that deals with regulation of and assessing of risks associated with carcinogens, chemical and ionizing radiation. And you have a threshold model that deals with every other type of response that acts, in theory, via a non-cancer means. And so these two models have basically ruled the biological, pharmacological, and regulatory world. However, the hormetic model somehow began to take some interest in the 1980s, renewed interest in the 1980s for a variety of reasons. And actually, it wasn't just the hormetic use of the hormetic term. Many other scientific disciplines started reporting in the literature biphasic dose responses, U-shaped dose responses, things that were coming in from pharmacology, from epidemiology, from basic biochemistry, and the toxicology world. And however, there was a wide range of terms that were used. I got involved around 1985 with this, and I've kind of continued it to the present time, although I'd have to say that my first introduction to the world of biphasic dose responses occurred when I was a young undergraduate in 1966, in which I was able to do a study with a synthetic growth inhibitor with plants, and found that this inhibitor at low doses stimulated the growth of peppermint plants at low doses, inhibited at high. My advisor never believed it, forced me to replicate the experiment in progressively stronger ways 8 different times. And then he finally became essentially grudgingly to the point where he accepted the reproducibility of the findings. He then had me do the work in hydroponics, not in soil. We got the same results with many replications. And the work was published, and And I never knew the term hormesis. I called it a low-dose stimulation and a high-dose inhibition, but in retrospect, it was an example, my first example of hormesis when I was a 20-year-old student, put it that way. Now, in the course of putting together hormetic dose responses, I was not sure how generalizable this was. I knew that it could occur. I'd seen it with my own eyes and my own experience, but I didn't know what the quantitative features might be. I didn't know if it could be extended across all biological type organisms, across all endpoints, chemicals, ionizing radiation. I really had no idea, but I started to grapple with this and develop criteria for its evaluation, and began to learn and tried to understand, in fact, why this was ignored by so many people for so many years. Now, what I found is that I have now about 5 different hormesis databases. I have about 70,000 examples of hormesis in these different databases. Each database is created for a different reason. But what I found is that the hormetic dose response is in fact very unique in general in its presentation, because if you find a hormetic dose response in plants and microbes, invertebrates, vertebrates, whole organisms, and humans, for in vivo, in vitro, different levels of biological organization, at the level of the cell, at the level of the organ, at the level of the whole organism, you find that the quantitative features of the dose response are really basically the same. It's a modest overcompensation. We'll see slides to this effect, where the maximum increase is very limited. It's only about 30 to 60% above the control. This was very surprising to me. I had no idea that this was actually the case. And then another surprise was that the width of the stimulation was typically about 10 to 20 fold. But then we found examples in which the stimulatory zone could extend out beyond a factor of 1,000. And this wasn't trivial. It occurred in 5 to 10% of our examples. So, but even in those cases, it was still a modest stimulation, but you could have a more variable zone of stimulation, and that had potentially some very interesting clinical implications. Now, people get hung up, especially in the environmental world, over whether that low-dose stimulation is actually good for you or bad for you. You know, in the world that I come from, which is really an environmental toxicology world, there's great concern with the hormetic dose response, because what they'll say is if you see a low-dose stimulation for arsenic or cadmium or lead, things of that nature, then, oh, Calabrese, you're saying that a low dose of lead is good for you. Or you're saying a low dose of arsenic is good for you. Or you're saying, oh, if you smoke 3 or 4 cigarettes a day, that's the hormetic dose. Is that what you're saying? It may well be what I'm saying, but basically I think that what I'm trying to say is first to look at this from a scientific perspective, and that is there a reality of a biphasic dose response? Can you, in fact, reproduce that phenomenon and leave the interpretation to a second level evaluation? Sometimes, in fact, you'll find that low dose exposure is not in your best interest. For example, we've looked at thousands of dose responses with anti-tumor drugs from the U.S. National Cancer Institute, and found that, in fact, most of the anti-tumor drugs that were effective in killing tumor cells at low doses actually stimulated the proliferation of the tumor cells in a biphasic, hormetic sort of way. So in fact, the concept of hormesis is really neutral. It's value neutral. and it should be seen as it's scientific. But it has implications that are important for environmental regulation, for the biomedical community, the therapeutic application. But sometimes that low-dose stimulation may be desirable, and sometimes that low-dose stimulation may in fact be undesirable. And you have to know the difference between the two. The interesting thing is that the magnitude of the stimulation is the same, more or less, whether it's beneficial, the term beneficial, or termed harmful, or in fact that low-dose stimulation could be neutral, may not have any clinical implications or environmental implications. Now, typically what we see here is this is a schematic of a situation in which you have an anti-tumor drug being applied to a tumor cell line. And you can see that at high doses, the therapeutic zone, it actually kills the tumor cell. But at low doses, you get the enhanced growth, and it shows an inverted U-shaped hormetic-like curve. Here we have a similar hormetic-like curve, but this time we're dealing with increasing memory. Now, drugs that improve memory and performance that way, retention, would be viewed as beneficial at low dose, and would be non-desirable if, in fact, at high dose they reduced memory. So you could have a hormetic response in one case which is not desirable, and then in another case in which it's desirable, and the researcher and the clinician has to know the difference here. Now, how do we assess hormesis? We study dose-response patterns, statistical significance. We try to replicate findings much more than a normal situation, because this limited stimulation of 30 to 60% could easily be due to chance. And you could easily mislead yourself to think that it was real and it was a hormetic response, when in fact it just may be variability. So what you really need to have is you need to be very, very tough-minded and have very strong study designs with enough doses, especially in the low-dose zone, to evaluate the consistency of a low-dose stimulation response. And then you actually have to replicate that a number of times before you can begin, I think, to have confidence in the findings, because we're really talking about something that is not threefold, fourfold, fivefold greater than the control value. We're talking about increases in the percentage zone. Now, you may think that this is unusual, but it's not. This is what pharmacologists have to deal with all the time when they're trying to improve biological performance of any different system. Because what we're going to find in the hormetic situation is that the hormetic dose response, which is a limited dose response but very generalizable, actually what that 30 to 60% range is telling me, and I hope will tell you, and that is actually defining the biological plasticity of biological systems across the entire spectrum of life, from plants to all type of animal life. And that is that all systems have a capacity to increase performance. And by performance, what do I mean? I mean, you could increase longevity, you could grow hair, you could strengthen bone, you could have disease resistance. But these changes that we see in hormetic systems are all limited to this 30 to 60%. And what that actually tells me is that as physicians, as people trying to develop new drugs, the most you're going to get out of a drug is really 30 to 60%. And that's basically, I think, what nature has told me. We also study mechanisms and try to document them. We do simulation studies with statisticians who essentially try to estimate the capacity of variability to falsely show positive findings. And so we try to be very rigorous using simulation studies. So in general, I have a number of databases, many thousands of dose responses that are indicative of this hormetic dose response using very rigorous entry and evaluative criteria. Now, in my opinion, hormesis is very general. It's independent of model. You see it in the plant world, the microbial world, the invertebrate world, the vertebrate world with humans. As I mentioned before, in vivo, in vitro. It's independent of endpoint as well. Agent and the level of biological organization. Something as important as this, it's really hard to imagine how the scientific community could have missed a fundamental concept until the year 2000, more or less. I mean, in the question and answer period, I'm sure I'll address how that actually occurred. But how is it possible for all these smart people in the toxicology, biology, pharmacology, and other related worlds to miss a fundamental concept until the year 2000, more or less? Well, we talked about the dose response being modest and not greater than 30%, and that's maybe one of the contributing factors. We talked about the stimulatory width before, and this is the standard curve that I typically show, that you have a low-dose stimulation that essentially is 30 to 60% greater than a control line, but you see that here, with a width of about 10 to 20-fold. Now, when you come to giving lectures on toxicology, you can show many different examples of hormesis, hundreds, thousands if you'd like. But basically, really rigorous people say, well, what's the mechanism? How does it actually work? Okay, well, we do have many mechanisms now for hormetic dose responses, and each mechanism is actually unique to the model. There's no single hormetic mechanism. And it's also unique to the tissue, endpoint, and agent. And I'll give you some examples here, but people look at receptor biochemistry, opposing receptors leading to stimulatory pathways or inhibitory pathways. Receptor desensitization has also been used. But in the practical world here, how you can really show a hormetic mechanism is if you have a biphasic dose response and you think it's mediated by a certain receptor, and you have an antagonist for that receptor, you can go in and apply the antagonist in the experimental model, and the antagonist essentially, if that hormetic response is mediated by that receptor pathway, you can block it and flatten that stimulation right out. And sometimes you can actually show that the inhibition is also mediated by the same receptor, and it flattens the entire dose response out. Sometimes it doesn't do the inhibition, but I have about 200 examples, very clear examples in the literature in which people have had biphasic dose responses using receptor antagonists in which they have gone directly in and they have blocked the receptor, and as a result, they have blocked the hormetic stimulation. It's, to me, clear proof that stimulation is mediated by that receptor. Now, there's more to the mechanism than that, but people do then go deeper into the mechanism as well. Okay, I've mentioned, and also, I have about 200 dose responses where people have gone into cell signaling processes and using certain types of receptors for these cell signaling processes. Likewise, blocked a biphasic dose response, remove the inhibition, the response returns, and gone into this at the level of both stimulation and inhibition. So regardless of the mechanism, this is very interesting. Whether it's a cell receptor, it's cell signaling pathway, a non-receptor mediated, it's compensatory or direct stimulation, the quantitative features of the dose response are similar. And this keeps coming back. Is it regardless of what the mechanism is, the hormetic dose response is actually a constant that you see. And you might say, how is this possible? It must be very important and significant to have been conserved evolutionarily from across plants all the way up to humans, that they have selected for and maintained a similar dose-response strategy. And I believe that the reason why this is the case is that it's a biological resource management issue, that you actually have to control your resources when you upregulate. If you essentially have no controls over what the regulation is going to be, then you can really waste your resources. And I believe that what's happening here is that there is a control over essentially how much biological collateral or resources that you can apply to a certain situation. Now, it's also important to know that hormetic responses can depend and can be opposite, depending upon the physiological state of the organism. For example, I have seen many studies with arthritic animal models treated with low doses of radiation and high doses of radiation. And in these situations, these arthritic models actually can have their arthritic symptoms essentially greatly alleviated at low dose and then exacerbated or made worse at higher dose. And what actually happens in these cases is that the ionizing radiation is actually inducing an anti-inflammatory phenotype. And the mechanisms have been worked out to explain this. However, if you gave the same doses of ionizing radiation to the same organisms in a non-inflamed state, what you would do is actually you would stimulate rather than dampen down the immune system, and you would stimulate it in a hormetic fashion with an inverse U-shaped dose response. So the actual response is hormetic in both situations, but it's opposite. It's a very interesting phenomenon, and it's dependent upon the physiological state of the organism. In my opinion, when you're studying hormesis, I see hormetic responses as being integrative responses across multiple levels of biological organization. For example, when I study hormetic situations, I tend to see examples with respect to cell proliferation, with respect to fecundity, with respect to tissue repair, with respect to behavior and learning, with respect to disease and injury resistance, in the context of, and also in the context of pre and post conditioning. I see it in aging and longevity. These are all very complicated processes. They involve hundreds, maybe thousands of supportive elements that go into them. When you take a look at the signaling, cell signaling that goes into any of these, the cell signaling is complicated, and there are many stages, but they actually lead to the formation of an integrative response. And it's the integrative response that the hormetic concept is applied to. If you were to look at the magnitude of the signal in a cell signaling system, it may go up 3-fold to 7-fold, or maybe more than that under some occasions. But that's a signaling of a phosphorylation, a molecule under certain circumstances and others. But the integrative response is what I'm talking about here. Some examples that I'm going to show you now are selected just to give you a broad range. Here's an example of acute ethanol exposure on social behavior in adolescent rats tested when they were 30 days old. And you can see that there's a low-dose stimulation. They become very active and playful. And at higher doses, their behavior is markedly diminished. Here you have an example in which rats were studied to try to figure out if you could reduce their anxiety, because rats do not like to spend time in lighted areas. They like to spend time in darker areas. And the particular agent that's being tested here, as you can see, at low doses, it actually leads to more time being spent in the lighted areas. At higher doses, it actually increases the time in the lighted areas and actually increases anxiety. And so from a pharmacological point of view, these doses here seem to be ideal. And in fact, in the pharmacology world, you can go from this type of preclinical study that can support the efficacy of a drug and then get it into a clinical trial. Here you have the effects of different doses of morphine on seizure threshold using an animal model that's induced by a particular chemical with the abbreviations PTZ. Now, the interesting thing here is that if you can increase the dose necessary to induce seizures by PTZ, it suggests that you have a good potential chemical for essentially people who have different types of seizures. Now, here you can see a nice dose response with lots of doses, and you see a nice peak at around 30% above the control, and then it goes below the control at higher doses. When I looked at this, that's an example of hormesis. And then I looked at about 70 to 100 other cases of drugs that in the preclinical area that we're looking at trying to detect whether these drugs might be effective in treating seizures. And I'd have to tell you that all of them showed this inverted U-shaped dose response with the same type of quantitative features, all displaying hormetic dose responses. Yet I would claim to you that nobody who was working in the pharmaceutical world who was making use of those data, who developed those data, in fact, ever called it hormesis. They probably said, oh, we have a low-dose stimulation. Oh, we have a high-dose inhibition. Oh, it's 30 to 60% above the control. And that's all they know. And they use it in the next level of clinical study, but they don't know that that's what actually happens with anxiolytic drugs. They don't know that's what happens with plant growth. They don't know that that happens with many other situations. They have missed the fact that the hormetic dose response is actually a basic biological principle. Here we have the effects of methanol on fruit fly longevity. And you can see, again, this is a nice inverted U-shaped dose response, but this is a very reproducible finding by investigators in the field. Here you have sodium arsenate on lymphocyte stimulation in bovine lymphocytes. Again, many doses showing this inverted U-shaped dose response. Here we have an example in which I'm going to try to show you how people have done some mechanistic work. Here we're looking at corticosterone on prolactin production, and you have a nice inverted U-shaped dose response. But we're now going to apply a corticosterone receptor antagonist to the experimental system. And what happens when you do that? Well, this stimulation, which looks pretty nice here, actually flattens right out across the entire dose response framework. So what is actually happening here is that the chemical corticosterone is actually having its stimulatory response as well as its inhibitory response mediated by this receptor. So it gives you an insight into the mechanism behind this particular biphasic dose-response relationship. Here's an example of quercetin on proliferation of estrogen-positive and estrogen-negative cell lines. You have two negative cell lines here, and you can see their dose response is in fact somewhat of a threshold-like. But in the case of the estrogen-positive cell lines, you can see at some point there's a low-dose stimulation and then a high-dose inhibition. Here we have an example of data at the high end that was analogous to what was used by Hermann Muller when he won his Nobel Prize in 1946 for being the first one to show that an environmental factor could cause mutation. He showed that X-rays could in 1927. And at high doses, X-rays do, in fact, cause mutations to the germ cells of Drosophila. But one thing that Muller did not do was he didn't use low doses. Later investigators replicated what Muller did, but then decided to use very low doses. And guess what? If Muller had used low doses, he not only would have shown that radiation could cause mutation, he would also show that at low doses, you actually reduce the background rate and get this J-shaped dose response that is very reproducible. And so this particular idea of using low doses has been ignored and dismissed and marginalized because either people emphasized high-dose effects or because, in fact, at least one of the other reasons was that the difficulty in actually doing appropriate studies where you have enough, a strong study design with enough doses and enough replication to do this. In the world of toxicology that I come from, our national toxicology program is based upon what? It's based upon high doses and few doses. Okay? What's a high dose? The maximum dose that the animal can tolerate for its whole life without losing more than 10% body weight. And then 1/2 of that. That's what our standards were based upon. They've now added a third dose to make it, 1/4 of the total. They're massively high doses. And so when you have massively high doses, what's the chance of finding a hormetic effect? Okay, it would be like me saying, what's the chance of me finding a desert in the ocean? I mean, you have to, if you don't look in the right place, you're never going to find it. Here's another example of cadmium, and I think the time is running late here, but the cadmium has an inhibitor. Do I have to stop? Oh, sorry. Okay. Over here, cadmium has a common inverted U-shaped dose response on human embryo lung fibroblast cell proliferation. It's a very reproducible response. People have looked at the mechanism of this, and they find that it acts, and it's controlled by a certain cell signaling pathway that involves the ERK pathway. If you take an inhibitor of that pathway, you markedly reduce that cell proliferation, essentially showing that low-dose stimulation actually is mediated by a certain pathway, that it's real, that it's reproducible, and you have a mechanism. Okay, see if I can get back over here. Over here you have inverted U and a J-shaped dose response for different endpoints, proliferation and apoptosis with arsenic. Now what we find here is a JNK pathway inhibitor markedly blocks the increase in cell proliferation and decreases apoptosis. The ERK inhibitor partially blocks this. And what it's really telling us is that this process is really generally dominated by the JNK pathway, but there's some crosstalk between that and the ERK pathway. But the point here is that when you find hormetic mechanisms, they play into the basic understandings of pharmacology and toxicology and molecular mechanisms. And there are molecular mechanisms, a lot of them. On the application side of hormesis, essentially can be universally applied. In the drug world, we see it for anxiolytic drugs, and I would say that essentially every anxiolytic drug that's on the market today, and many that never made it to the market, the animal models were biphasic, and they picked off the doses in the hormetic zone. And they never called it hormesis, they called it biphasic. Anti-seizure drugs, the same situation. Memory enhancement, the same. It's almost always an inverted U-shaped dose response. The six drugs that have been approved by the U.S. FDA for treatment of Alzheimer's disease in the U.S. in their preclinical data all show inverted U-shaped dose responses for memory retention. Osteoporosis, the same thing. I have a vast literature on wound healing and acceleration by many different agents, endogenous, exogenous agents, almost all showing acceleration following a hormetic dose response. I could apply this now to the plant world and try to increase production. I could apply it to a lot of other different areas. The hormetic concept is a general one. It's I believe, a general principle, and it has application, but it also is not a free ride for the industry, because it's very difficult when you are in the pharmaceutical world and the best you can hope to get out of a system is 30 to 60%. And that's very hard to even prove when you have a clinical trial with highly heterogeneous human populations, trying to show a 30%, 40% increase when your background variability could be 10, 15, 20%. You pull all the data and you don't show efficacy, and you've lost millions of dollars of investment. That's a very difficult thing for a pharmaceutical company to deal with, especially when your preclinical data have been very good and very clean. And why are they very good and very clean? Because your animal model has far less variability. It's very homogeneous, and you can detect that nice increase. But when you apply the drug to a highly heterogeneous population, where they all have their own individual dose responses, it's very difficult to show efficacy, and I believe that's why many drug companies have looked to personalized medicine to try to recoup this, because they have excellent drugs based on preclinical trials, fail oftentimes in the clinical trial, and yet they know that these drugs can work with some people, or they believe they can, and they can target it. Also, you might have heard of pre- and post-conditioning. Preconditioning is very significant in the biomedical world, where a prior stress can protect you against a subsequent more massive condition. And that is first discovered in 1986, giving a dog a very low hypoxic stress to the heart, and then 2 days later giving it a massive myocardial infarction and reducing the damage by some 70 or 80%. Well, this phenomenon has now been generalized to many different models and to humans, and generalized also to many different tissues. Now, the interesting thing here, and why I'm relating it, is that if you take many different small but progressively larger preconditioning doses, many different ones, and then you do the study, you will find that the dose response follows a beautiful inverted U-shaped dose response, totally consistent and explainable by what? By the hormetic dose response process. Preconditioning and postconditioning are really manifestations of hormesis, further supporting the generalizability of this concept. Now, people also talk about many different drugs. What if I give 2 drugs together, 3 drugs together? Can we boost this response? Well, in my opinion, I don't believe you can. If you want to boost memory beyond, you have a drug that increases it by 30%, you have another drug in the same model that increases it by 30%, you combine these drugs, you're going to get 200%, 120%. I believe from the data that I've seen that it caps off at this hormetic constraint. And that's a very significant issue. People in the world, you've heard of metabolic engineering. Well, people in the world of metabolic engineering are trying to break the bounds of plasticity by adding 1,000, 2,000, slices of a gene for a particular, particular activity, and other ways to try to have metabolic engineering. They've been at this game for 20-something years, and it's very difficult to do this because the biphasic dose response has redundant controls in it. And we don't even understand the nature of all those redundant controls and how they work, and metabolic engineering is trying to tease those apart. I hope they can, because we will benefit from it, but this is not an easy situation. Okay, so the low-dose stimulation is different than high-dose toxicity. And in my opinion, what is the low-dose stimulation? In my opinion, what the low-dose stimulation is actually telling us is that it's a measure of biological performance. It's what you can get out of the system. So that has great relevance to the pharmaceutical world and to physicians, because in addition to trying to prevent people from getting sick by killing bacteria or knocking out viruses or other things, you're actually trying to improve their performance in a lot of different ways. Strengthening bone, strengthening memory, lots of things, resisting diseases. So you have, there's a complexity here to it, but you're trying to improve performance. But how much can you get out of the system? Well, Hormesis decides how much you can get out of the system. Hormesis is the boss. In my opinion, Hormesis is the first quantitative estimate of biological plasticity. In my opinion, Hormesis, the Hormesis stimulatory response is constrained by the limits of plasticity. Now, there are many applications that we have, but I also have to tell you one thing historically. Obviously, I've been heavily involved with the Hormesis dose response for, when I had brown hair and now I have gray hair. So it's kind of followed me through my adult life. And what I've seen is when I first, around 1999, 2000, when I was really putting a lot of things together, I said, well, I wanted to validate the hormetic model in some sort of way and test its frequency in the population in studies. And so I said, well, how did the government and how did the scientific community validate the threshold model? Because I'll just copy how they did that. I never validated a model before. So just find what they did, follow what they did, maybe I'll do it right. So I looked and 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 I looked, and guess what? I never found that any government, any scientist, any industry, any expert committee ever published a paper in which they attempted to or showed validation of the threshold model for making accurate predictions below the threshold. Never. And we live below the threshold. All of our standards were based upon a model that was never validated by anyone. Okay? All the drugs, all the environmental contaminants that are based upon the threshold model have a model that was never validated, never thought to be validated. So I decided to try to validate it. And so what happened is I got 3 or 4 very large data sets looking at a wide variety of compounds. Applied statistical analysis with a team of researchers to test the linear, threshold, and hormetic dose responses. And guess what happened? In every single case, the threshold and the linear model failed to make accurate predictions in the low dose zone. And the hormetic model was the only one that actually did it. We submitted it to the leading toxicology journals, went through some tough peer reviews, and all 3 made it through. And basically what we showed for the very first time was that, in fact, the models that are used by the regulatory agencies fail when put to the test. The only one actually that passed the test was the one that the regulatory agencies actually ignored, marginalized, and basically would never consider. And so what you have is you have a world that is governed by non-science, really. And it's hard to tell them, it's hard to tell the teacher that they're wrong. So in conclusion, Hormesis, in my opinion, is general and central. It affects all disciplines utilizing the dose response. It represents a general adaptive strategy through which biological and biomedical performance is enhanced and mediated. The hormetic responses will often govern efficacy and hazard risk assessment for pharmaceutical and chemical products. Product evaluation needs to consider the entire dose response in order to enhance product and drug discovery and development and clinical evaluation. Thank you very much.